Now in this lesson we are going to see the graphical representation of the C2 cycle that we had talked about earlier. Now you're going to see that what happens in which organelle, that is what I have tried to represent over here. Now what you see over here is that this particular graphical representation has been divided into 3 zones, and those zones represent 3 different organelles. The first being the chloroplast. As you can see, this particular box represents the chloroplast. Then the second is peroxisome, though keep one thing in mind, that in actual conditions they would not be arranged like this. Just in order to represent, and in a cyclic form I have used the representation where three of them are lying in close proximity to each other, and the third one is mitochondria. Now as we had talked about that when one molecule of RUBP combines with oxygen, when Rubisco acts as oxygenase instead of carboxylase, it gives rise to one molecule of phosphoglyceric acid, which is shown over here, and one molecule of phosphoglycolate, which is a 2-carbon compound that is being formed. So we are to see in this lesson that what happens to phosphoglycolate. Try to recall what all reactions we had studied in the previous lesson. Those reactions would be discussed over here. See, when this RUBP combined with oxygenase activity showing Rubisco and oxygen, it gave rise to a 2-carbon compound called phosphoglycolate and a 3-carbon compound, that is 3-phosphoglyceric acid, which would undergo the same reactions as we had studied earlier. That is a different course altogether. We are to confine our studies to this 2-carbon compound. What happens to that? Now as you can see over here, this phosphoglycolate would get converted to glycolate within chloroplast itself, and this glycolate would move to the peroxisomes, where it would make sure that a peroxide compound is formed when it gets converted to glyoxylate. Alright, when this glyoxylate is converted to glycine, the process is of deamination. This glycine moves to mitochondria, where it loses one of its carbon dioxide and uses NAD positive, forms NADH, leading to formation of serine. This is an amino acid. All these are amino acids. Here it happened to be deamination. When this deamination was taking place, there was a coupling reaction which was taking place, that glutamate was converting to alpha ketoglutarate. Okay, this coupling reaction was taking place inside the peroxisomes when phosphoglycolate, which got converted to glycolate, came inside the peroxisomes. It formed glycine by the process of deamination, but previously it had formed glyoxylate. That's why it was possible to form glycine. This glycine, on reaching the mitochondria, gets rid of one of the carbon dioxide. So you can see, instead of fixing the carbon dioxide over here, what is happening? One carbon dioxide is getting lost. And then the second stage is that this glycine, when it loses its carbon dioxide, it loses its ammonia as well. One molecule of ammonia is also lost, and it gets converted to serine. This ammonia, which is formed in mitochondria, comes and gets coupled with serine when it goes to peroxisomes. Okay, the serine has to go back to peroxisome. This ammonia in combination with serine and a coupling reaction that is taking place, where alpha ketoglutarate is getting converted to glutamate. This is going on. This serine gets converted to hydroxypyruvate, because the ammonia from the release of glycine getting converted to serine is being used up in the formation of hydroxypyruvate. Now the next conversion would be that this hydroxypyruvate within the peroxisomes would get converted to glycerate. Now this glycerate, when it is getting converted, there is another reaction. The NADH is getting converted to NAD positive. This glycerate would further move to chloroplast, getting back to chloroplast, and this glycerate would get converted to phosphoglyceric acid by using 1 ATP. So this is all the reaction is all about. This is just the graphical representation. I am taking as a given that you have a good idea about photorespiration that we talked about. Now in this particular lesson we have seen that how all this reaction is taking place in different, with context to different organelles that we have discussed. And one thing, as you can see over here, one of the carbon dioxide is getting lost from whatever compound is being utilized, and one of the ATPs is also being used, and we are nowhere getting glucose. So this is a wasteful process. That is why it is expected from Rubisco that instead of acting like oxygenase, it should act as carboxylase, and that is why the process of photorespiration that we have discussed in this lesson is a wasteful process. Do keep in mind. Why? Because there is no fixing of carbon dioxide that is taking place. Whatever was generated is being used, and nothing, concrete is being formed. Now, what is concrete for a plant? It is the glucose molecules which would be formed. So, we are not getting any glucose molecule. There is no fixation of carbon dioxide. It is getting evolved instead. The oxygen which was responsible for bringing about this change by, you can say, bribing the Rubisco, has given rise to nothing substantial which would be useful, because we are discussing photosynthesis. There should be synthesis of glucose, and this is photorespiration process which is totally wasteful for the plant. So this is what we have studied. Now, because this is a wasteful process, next what we are going to study is that plants have coined a process which is responsible for avoiding this particular wasteful process. So the next lesson would be dedicated to another type of photosynthetic process, that is again the dark reaction, and that would be called as C4. For that, watch the next lesson.